I admit that I'm a little bit nervous here because I'm going to say some pretty radical things about how we should think about cancer differently to an audience that contains a lot of people who know a lot more about cancer than I do. But I will also confess that I'm not as nervous as I should be because I'm pretty sure I'm right about this. And that this, in fact, will be the way that we treat cancer in the future. In order to talk about cancer, I'm going to actually have to, let me get the big slide here. First, I'm going to try to give you a different perspective of genomics. I want to put it in perspective of sort of the bigger picture of all the other things that are going on. And then talk about something you haven't heard so much about, which is proteomics. Having explained those, that will set up for what I think will be a different idea about how to go about treating cancer. So let me start with genomics. It is the hot topic. It is the place where we're learning the most. It's the great frontier. But it has its limitations. And in particular, you've probably all heard the analogy that the genome is like the blueprint of your body. And if that were only true, it would be great, but it's not. It's like the parts list of your body. It doesn't say how things are connected, what causes what, and so on. So if I could make an analogy, let's say that you were trying to tell the difference between a good restaurant, a healthy restaurant, and a sick restaurant. And all you had was the list of ingredients that they had in their larder. So it might be that if you went to a French restaurant and you looked through and you found they only had margarine and they didn't have butter, you could say, I see what's wrong with them. I can make them healthy. And there probably are special cases of that. You could certainly tell the difference between a Chinese restaurant and a French restaurant by what they had in a larder. So the list of ingredients does tell you something, and sometimes it tells you something that's wrong. If they have tons of salt, you might guess they're using too much salt or something like that. But it's limited because really to know if it's a healthy restaurant, you need to taste the food. You need to know what goes on in the kitchen. You need the product of all of those ingredients. So if I look at a person and I look at a person's genome, it's the same thing. The genome really, the part of the genome that we can read is the list of ingredients. And so indeed, there are times when we can find an ingredient that's bad. Cystic fibrosis is an example of a disease where you just have a bad ingredient and you have a disease, and we can actually make a direct correspondence between the ingredient and the disease. But most things, you really have to know what's going on in the kitchen, because mostly sick people used to be healthy people. They had the same genome. So the genome really tells you much more about predisposition. So what you can tell is you can tell the difference between an Asian person and a European person by looking at the ingredients list. But you really, for the most part, can't tell the difference between a healthy person and a sick person, except in some of these special cases. So why all the big deal about genetics? Well, first of all, it's because we can read it, which is fantastic. It is very useful in certain circumstances. It's also the great theoretical triumph of biology. It's the one theory that the biologists ever really got right. It's sort of fundamental to Darwin and Mendel and so on. So it's the one thing where they predicted a theoretical construct. So Mendel had this idea of a gene as an abstract thing. And Darwin built a whole theory that depended on them existing. And then Watson and Crick actually found one. So this happens in physics all the time, that you predict a black hole and you look out the telescope and there it is, just like you said. But it rarely happens in biology. And so this great triumph is so good that it's almost a religious experience in biology. And Darwinian evolution is really the core theory. So the other reason it's been very popular is because we can measure it. It's digital. And in fact, thanks to Kary Mullis, you can basically measure your genome in your kitchen with a few extra ingredients. So, for instance, by measuring the genome, we've learned a lot about how we're related to other kinds of animals, and by the closeness of our genome, or how we're related to each other, the family tree, or the tree of life. There's a huge amount of information about the genetics just by comparing the genetic similarity. Now, of course, in medical application, that is very useful because it's the same kind of information that the doctor gets from your family medical history, except probably your genome knows much more about your medical history than you do. And so by reading the genome, we can find out much more about your family than you probably know. And so we can discover things that probably you could have found by looking at enough of your relatives, but they may be surprising. I did the 23andMe thing and was very surprised to discover that I am fat and bald. But sometimes you can learn much more useful things about that. But mostly, what you need to know to find out if you're sick is not your predispositions, but it's actually what's going on in your body right now. So to do that, what you really need to do is what's going on, you need to look at the things that the genes are producing and what's happening after the genetics, and that's what proteomics is about. Just like genomics is the study of all the genes, proteomics is the study of all the proteins. And the proteins are all of the little things in your body that are signaling between the cells, actually the machines that are operating, that's where the action is. Basically, a human body is a kind of a conversation going on both within the cells and between the cells, and they're telling each other to grow and to die, and when you're sick, something's gone wrong with that conversation. And so the trick is, unfortunately, we don't have an easy way to measure these like we can measure the genome. So the problem is that measuring it, if you try to measure all the proteins, it's a very elaborate process. It requires hundreds of steps. And it takes a long, long time. And it matters how much of the protein it is. It could be very significant that the protein changed by 10%. So it's not a nice digital thing like DNA. The basic problem is somebody's in the middle of this very long stage, they pause for just a moment, and they leave something in an enzyme for a second, and all of a sudden all the measurements from then on don't work. And so people get very inconsistent results when they do it this way. People have tried very hard to do this. I tried this a couple of times and looked at this problem and gave up on it. I kept getting this call from this oncologist named David Agus. And Applied Minds gets a lot of calls from people who want help with their problems, and I didn't think this was a very likely one to call back, so I sort of kept on getting on the delay list. And then one day, I get a call from John Doerr, Bill Berkman, and Al Gore on the same day saying, return David Agus' phone call. So I was like, okay, this guy's at least resourceful. So we started talking, and he said, I really need a better way to measure proteins. I've looked at that, been there, no, it's not going to be easy. It's like, no, I really need it. I mean, I see patients dying every day because we don't know what's going on inside of them. We have to have a window into this. And he took me through specific examples of when he really needed it, and I realized, wow, this would really make a big difference if we could do it. And so I said, well, let's look at Applied Minds has enough sort of play money that we can go and just work on something without getting anybody's funding or permission or anything. So we started playing around with this. And as we did it, we realized this was the basic problem, not taking the sip of coffee, that they were humans doing this complicated process, and that what really needed to be done was to automate this process like an assembly line and build robots that would measure proteomics. And so we did that, and working with David, we made a little company called Applied Proteomics eventually, which makes this robotic assembly line, which in a very consistent way measures the protein. I'll show you what that protein measurement looks like. Basically, what we do is we take a drop of blood out of a patient, and we sort out the proteins in that drop of blood according to how much they weigh, how slippery they are, and we arrange them in an image. And so we can look at literally hundreds of thousands of features at once, but out of that drop of blood. And we can take a different one tomorrow, and you will see your proteins tomorrow will be different. They'll be different after you eat or after you sleep, or they really tell what's going on there. And so this picture, which looks like a big smudge to you, is actually the thing that got me really thrilled about this and made me feel like we were on the right track. So if I zoom into that picture, I can just show you what it means. We sort out the proteins from left to right is the weight of the fragments that we're getting. And from top to bottom is how slippery they are. So we're zooming in here just to show you a little bit of it. And so each of these lines represents some signal that we're getting out of a piece of a protein. And you can see how the lines occur in these little groups of bump, 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 bump. And that's because we're measuring the weight so precisely that carbon comes in different isotopes. So if it has an extra neutron on it, we actually measure as a different chemical. So we're actually measuring each isotope as a different one. And so that gives you an idea of how exquisitely sensitive this is. So this is sort of like, seeing this picture is sort of like getting to be Galileo and looking at the stars and looking through the telescope for the first time. And suddenly you see, wow, it's way more complicated than we thought it was. But we can see that stuff out there and actually see features of it. So this is the signature out of which we're trying to get patterns. So what we do with this is, for example, we could look at two patients, one that responded to a drug and one that didn't respond to a drug, and ask what's going on differently inside of them. And so we can make these measurements precisely enough that we can overlay two patients and look at the differences. So here we have Alice in green and Bob in red. We overlay them. This is actual data. And you can see, mostly it overlaps and it's yellow, but there's some things that just Alice has and some things that just Bob has. And if we find a pattern of things, of the responders to the drug, we see that in the blood. They say they have the condition that allows them to respond to this drug. We might not even know what this protein is, but we can see it's a marker for the response to the disease. So this already, I think, is tremendously useful in all kinds of medicine. But I think this is actually just the beginning of how we're going to treat cancer. So let me move to cancer. The thing about cancer, when I got into this, I really knew nothing about it. But working with David Agus, I started watching how cancer was actually being treated and went to operations, where it was being cut out. And as I looked at it, the way I realized, to me, it didn't make sense how we were approaching cancer. And in order to make sense of it, I had to learn sort of where did this come from. We're treating cancer almost like it's an infectious disease. We're treating it as if it's something that got inside of you that we have to kill. So this is the great paradigm. This is another case where a theoretical paradigm in biology really worked, was the germ theory of disease. So what doctors are mostly trained to do is diagnose, that is, put you into a category, and apply a scientifically proven treatment for that diagnosis. And that works great for infectious diseases. So if we put you in the category of you've got syphilis, we can give you penicillin. We know that works. If you've got malaria, we give you quinine, or some derivative of it. And so that's the basic thing doctors are trained to do, and it's miraculous in the case of infectious diseases how well it works, and many people in this audience probably wouldn't be alive if doctors didn't do this. But now let's apply that to systems disease like cancer. The problem is that in cancer, there isn't something else that's inside of you. It's you, you're broken. All of those, that conversation inside of you got mixed up some way. So how do we diagnose that conversation? Well, right now what we do is we divide it by part of the body. Where did it appear? And we put you in different categories according to what the part of the body, and then we do a clinical trial for a drug for lung cancer, and one for prostate cancer, and one for breast cancer. And we treat these as if they're separate diseases, and that this way of dividing them had something to do with what actually went wrong. And of course, it really doesn't have that much to do with what went wrong, because cancer is a failure of the system. And in fact, I think we're even wrong when we talk about cancer as a thing. I think this is the big mistake. I think cancer should not be a noun. We should talk about cancering. It's something we do, not something we have. And so those tumors, those are symptoms of cancering. And so your body is probably cancering all the time, but there are lots of systems in your body that keep it under control. And so to give you an idea of sort of an analogy of what I mean by thinking of cancering as a verb. Imagine we didn't know anything about plumbing. And the way that we talked about it, we'd come home and we'd find a leak in our kitchen, and we'd say, oh, my house has water. We might divide it, the plumber would say, oh, well, where's the water? Oh, well, it's in the kitchen. Oh, you must have kitchen water. No, that's kind of the level at which it is. Kitchen water, you know, well, first of all, we'll go in there and we'll mop out a lot of it. And then we know that if we sprinkle Drano around the kitchen, that helps. You know, whereas living room water, it's better to do tar on the roof, you know. And it sounds silly, but that's basically what we do. And I'm not saying you shouldn't mop up your water if you have cancer. But I'm saying that, you know, that's not really the problem, that's the symptom of the problem. What we really need to get at is the process that's going on, and that's happening at the level of the proteomic action. It's happening at the level of why is your body not healing itself in the way that it normally does? Because normally your body is dealing with this problem all the time, just like your house is dealing with leaks all the time, but it's fixing them, it's draining them out, and so on. So what we need is to have a causative model of what's actually going on. And proteomics actually gives us the ability to build a model like that. David got me invited to give a talk at National Cancer Institute, and Anna Barker was there. And so I gave this talk and said, why don't you guys, you know, do this? And Anna said, because nobody within cancer would look at it this way. But what we're going to do is we're going to create a program for people outside of the field of cancer to get together with doctors who really know about cancer and work out different programs of research. So David and I applied to this program and created a consortium at USC where we've got some of really the best oncologists in the world and some of the best biologists in the world from Cold Spring Harbor, Stanford, Austin, I won't even go through and name all the places, to have a research project that will last for 5 years where we're really going to try to build a model of cancer like this. We're doing it mice first, and we will kill a lot of mice in the process of doing this. But they will die for a good cause. And we will actually try to get to the point where we have a predictive model where we can understand when something when cancer happens, what's actually happening in there and which treatment will treat that cancer. So let me just end with giving you a little picture of what I think cancer treatment will be like in the future. So I think eventually, once we have one of these models for people, which we'll get eventually, I mean, our group won't get all the way there, but eventually we'll have a very good computer model, sort of like a global climate model for weather. It has lots of different information about what's the process going on in this proteomic conversation at many different scales. And so we will simulate in that model for your particular cancer. And this also will be for ALS or any kind of system neurodegenerative diseases, things like that. We will simulate specifically you, not a generic person, but what's actually going on inside you. And in that simulation, what we could do is design for you specifically a sequence of treatments. It might be very gentle treatments, very small amounts of drugs. We might be things like, don't eat that day, or give them a little bit of chemotherapy, maybe a little radiation. Of course, we'll do surgery sometimes and so on, but design a program of treatment specifically for you and help your body guide back to health, guide your body back to health. Because your body will do most of the work of fixing it if we just sort of prop it up in the ways they're wrong. We put in the equivalent of splints. And so your body will basically, has lots and lots of mechanisms for fixing cancer, and we just have to prop those up in the right way and get them to do the job. And so I believe that this will be the way that cancer will be treated in the future. It's going to require a lot of work, a lot of research from, there'll be many teams like our team that work on this. But I think eventually we will design for everybody a custom treatment for cancer. So thank you very much.